Hi friends, you are watching Absolute Biopharma and I am your host Pallavi. On this channel you will find everything related to the biopharma world like the technical content, various job openings, the interview questions that are asked in the various interviews and various trainings based on the biopharma world. So today's topic for this video is protein and its structure. Let's begin with peptides and proteins. What are peptide and proteins? If you see my last video, I have described what are amino acids. So proteins are made up of amino acids. When multiple amino acids join together, they form a peptide, and when multiple peptides are joined together, they form protein. If you see in the figure, when two amino acids are covalently joined by amide linkage, they form a peptide bond. So if two amino acids are joined together by a amide linkage, they are called dipeptide. If three amino acids are joined together, they are named as tripeptide. If very few amino acids are joined together, they will form oligopeptide. If many amino acids are joined together by peptide bond, they are called polypeptide. If we refer here many amino acids, that means they can be from 10 to 10,000, and when thousands of amino acid residues are joined together, they form a protein. Means the number of amino acids joined together should be more than 10,000. If you see in the figure, when there is a loss of water molecule from the each amino acid, they leads to the formation of the peptide bond. In the result, we will get the amino acid with the amine terminal and carboxyl terminal. Now let us see some of the protein structures. So when you look at a protein sequence, say, of insulin written on a paper, it's easy to think of amino acids as beads and the proteins as a pearl necklace. But the protein folds into incredibly complex three-dimensional structures as they are being synthesized. In part, that 3D folded shape determines the protein's function. Biologists have divided protein structure into four classes: primary, secondary, tertiary, and quaternary. Primary structure is the amino acid sequence. If the amino acids are joined in a series, they are named as primary structure. The secondary structure describes the protein's three-dimensional structure due to the interaction between the backbone atoms. The backbone is made up of all the atoms that connect one amino acid to the next, and it's the same in every protein. So this set of atoms gives rise to a few different structures, and the most common of these are the alpha helix and the beta sheet. So the most important thing about alpha helices and beta sheets is that they help stabilize the folded protein structure through a network of hydrogen bonds between the backbone atoms. In an alpha helix, the main chain amide hydrogen makes hydrogen bond with the carbonyl oxygen that is 4 sides away. So you can see in the picture, the main chain amide hydrogen is bonded through a hydrogen bond with the carbonyl oxygen. A beta sheet also involves hydrogen bonding in the backbone, but instead of resulting structure being helical, it's mostly flat sheets or sheets of amino acids. The structure results from several regions of protein, unlike the alpha helix that results from one continuous region. So beta strands are between 5 and 10 amino acid residues in the length. Adjacent beta strands are aligned so that hydrogen bond form between an amide hydrogen and a carbonyl group. So here you can see the hydrogen bond form between the amide hydrogen and a carbonyl oxygen. So the secondary structure helps stabilize the folded protein structure through a network of hydrogen bond between the backbone atoms. Now let's talk about tertiary structure, which describes the position of every atom in a protein in the three-dimensional space. This means we now have to consider how the amino acid chains contribute. Just like with the secondary structure, there are few different ways in which the side chains can interact with each other or the backbone to stabilize the protein. Last comes the quaternary structure. It describes how identical or similar protein subunits come together to form a larger protein. Here is the example of hemoglobin. You can see there are 4 subunits, 2 beta subunits, 2 alpha subunits. All the four subunits come together to form a larger protein called hemoglobin. Now let's understand the protein structure with some examples. So first is cysteine. The cysteine has the ability to form disulfide linkages. Disulfides are covalent bonds, so they are strong enough to be significant stabilizing force in the protein even if there is just one. Next is aspartate and lysine. So these are the structure of lysine and aspartate. Here is what they look like at the physiological pH. Those opposite charges form an ionic bond between the two amino acids, stabilizing force for protein structure. So finally comes the insulin, that is an example of quaternary structure. So I have been describing insulin as a single molecule, and for the most part it is. But at higher concentration, two insulin molecules come together to form an insulin dimer. And in the presence of two zinc atoms, 6 molecules of insulin can come together to form a hexamer. So for example, insulin is synthesized and stored as a hexamer, a trimer of insulin dimer. Insulin is active as monomer at its binding site. But when insulin hexamer releases monomers, the monomers have a strong tendency for dimerization. Various insulin analogs have been engineered to reduce the tendency to form insulin dimer so that insulin can become more available to receptor quickly after injection. Even polypeptides like insulin that only have 51 amino acids can be structurally complex. In any protein, all types of bond contribute to the stability of the final folded shape and to its function. In this video, I have described what is a protein and a peptide, various protein structure like primary, secondary, tertiary, quaternary, and the respective examples. So finally, I would like to say after protein is translated, additional modifications of the structure are possible that affect the structure and function. Thank you so much for watching this video, and if you like the video and you found the content is relevant, then do subscribe to my channel and share it with your friends and colleagues so that they can also get the advantages of my videos. If you have any other topics in your mind, then do comment in the comment section. Thank you and have a great day.